I'm Ben Raphael. I'm at Brown for the next 6 weeks at least. I guess I'm supposed to give some sort of biographical thing, but I'll just say we've done a lot of work. We've published lots of papers. You know, maybe in honor of Donald Trump, I'll say these papers are so beautiful. They're the most amazing papers ever. You know, you should read these papers. You'll cry with just cry at the beauty if you read all our papers. What I've been tasked to do is give you a tutorial talk on cancer genomics, and this is actually a very active area, a very exciting area. And for the short course, at least for this first week in the schedule, it's the only talk really on cancer genomics. We just have this one tutorial talk, no research talks, and so it's really quite surprising in terms of the organization. So I'll definitely have to complain to the organizers about this oversight. So we've heard a lot about complex diseases, germline diseases. We haven't heard much about cancer, at least cancer as it relates to somatic mutations. And it's really somatic mutations, that is, mutations that are acquired, not as opposed to those you inherit from your parents, that drive cancer. Somatic mutations happen all the time. Every time a cell divides, it pretty much has some mutation in its genome, but most of them are benign or even slightly deleterious. But a small fraction of somatic mutations that occur give cells some selective advantage, might cause them to grow faster or be able to evade other normal controls. And through a progression of such somatic mutations and other changes in gene expression and epigenetic changes lead to the generation of a tumor. So, typical tumor, if we want to understand it's how it got to be, we'd like to know what these driver mutations are. But there's actually relatively few driver or causal mutations inside a tumor, and they're buried within the sea of passenger mutations. These mutations that are not really advantageous necessarily for the tumor, and just are ones that are there just from random chance. So, somatic mutations happen all the time. They happen from endogenous causes, just errors in DNA replication. They happen due to environmental exposures, UV light, for example, cigarette smoke, many others. And they happen on a range of scales, ranging from single letter, single nucleotide changes, all the way through large chromosomal rearrangements and aneuploidies. This means we have to detect mutations on these various scales. So just as one example, and perhaps the most famous example of a change in cancer is a translocation between chromosomes 9 and 22 that results in a fusion gene being created. This is the so-called Philadelphia chromosome. It was discovered in the late 60s, early 70s. And this fusion gene is between two genes, BCR and ABL, and actually drives the progression of CML and other types of leukemia. Interestingly, after decades of really detailed experimental characterization of this fusion gene, a drug, an inhibitor of this gene, came on the market about 15 years ago and has been highly successful in treating patients with this type of leukemia. So what this fusion gene is doing is it's activating the kinase, the ABL kinase, leading to all sorts of downstream changes through various signaling pathways in ABL. Downstream changes of downstream signaling changes from ABL, and then again, this drug is able to kind of target and inhibit this overactivation of this pathway. So I said it's been highly successful in treating patients with this type of leukemia. Unfortunately, what happens with many of the patients is that there are additional mutations that lead to resistance to this drug, imatinib or Gleevec. And these mutations can occur in BCR-ABL itself. They can occur by, say, amplifying BCR-ABL. They can occur in downstream pathways that are activated by the kinase. So there's all sorts of ways that the cell can get around this targeted treatment. And this leads us to sort of another issue that really overrides a lot of what we have to think about when we analyze cancer genomes, and that's that cancer is an evolving system. There's mutations that are accumulating. There's selection for mutations. By treating a patient with a drug, we might then change the selective pressure and thereby push the cells towards other mutational states. So this evolutionary process in a population of cells is part of what makes analyzing cancer genomes somewhat different from analyzing other complex diseases. So, if we wanted to maybe have a one-sentence definition of cancer, here's sort of my crack at doing it. It's a somatic evolutionary process that alters signaling and regulatory networks in the cell, sort of combining a lot of, what I just quickly went through with the BCR-ABL example. So what's new? This sort of idea, all of these ideas, this sort of picture comes from a review article by Peter Knoll, who was one of the discoverers of the Philadelphia chromosome. This review article is from like the mid-1970s. These are all very old ideas. What's new, and as you know, is that we can now measure genomes. We can measure them cheaply, we can measure them en masse, and so now we can start to quantify these processes, discover what are the driver mutations, discover what are the mutations that are responsible for the various phenotypes that cancer cells have. So the challenges are to some extent less in the data generation and more in the interpretation, which is where the computational approaches come in. There are large-scale projects. Many of you know probably the Cancer Genome Atlas, TCGA, that has characterized over roughly 11,000 samples from actually the sort of old. There are approximately 30 cancer types, parallel efforts with international consortiums. And these days, sequencing is cheap enough that even single institutions or even single investigators can generate reasonable-sized cancer cohorts. So TCGA had this feature of using multiple types of genomic, transcriptomic, and a couple of proteomic measurements of the samples. And this is important because it's really the integration of these multiple types of information that allow us to explain how the mutations in the genome lead to downstream expression changes, how methylation changes play a part in this as well. But for the purposes of this tutorial, again, given the short amount of time, I'm really just going to focus on this first layer, which is the mutation and copy number changes, so changes that are happening in DNA, and less on the transcriptome, although we've certainly had some tutorials on RNA-seq, and a lot of the methods that you heard about there are applied to cancer samples as well. And there are techniques that are now doing proteomics, and TCGA was an antibody-based technique called RPPA, but there's mass spectrometry-based techniques as well that are characterizing the proteome. So at the level of somatic mutations, I would say that the challenges, the sort of analytical challenges, are roughly can be divided in two. One is just sort of measurement, which is what are the somatic mutations present in the sample, and two, then interpretation. And this is classic genotype to phenotype, things that you would see in GWAS, but with some unique twist. In fact, both of these challenges, if I crossed out somatic and put germline mutation, then this would be exactly the same two challenges that are faced by essentially any GWAS. What makes cancer genomics and somatic mutations sort of different and presents some unique challenges is that tumors are very heterogeneous. And this heterogeneity happens on two levels. One is so-called intertumor heterogeneity, which is to say that if you look at any two individuals with the same cancer, same type of cancer, same even subtype of cancer, the somatic mutations in their tumors tend to be quite different. In contrast to sort of other complex disease genetics that's done at the germline level, somatic mutations occur independently in each individual, so we're not really as interested in inheritance. We don't have to correct for population structure, these challenges that you might have in GWAS. But the genetic architecture, like actually many germline diseases, is also very complicated. But heterogeneity actually happens at another level, which is that the tumor is a collection of cells. Those cells are growing and dividing, and new mutations are being acquired, and some of those are being selected for. And each then cell in the tumor might have different complements of somatic mutations. And most of the data that we have, and the vast majority of the sequencing that's being done, is being done on bulk samples. So if you're looking at germline data and you take a blood sample and you can measure someone's germline genome, and it's fine because essentially all you care about is the germline genome. When you measure a tumor, you might actually be missing information because the individual cells have different somatic mutations. So not every genome in the tumor is identical. In fact, they're all very different. But most of the data that we get gives us this mixture of cells. So what I wanted to discuss in this tutorial was sort of three topics. One is just sort of a quick blast through the mutation detection problem. We've heard a lot about this already, so I won't spend too much time on it. A little bit on tumor evolution, which is somewhat different from the types of things that you might see in other disease studies, and then finally a little bit on prediction of driver mutations and causal mutations. Now I should say, when I went to sleep last night after the Hollywood Bowl and after a few glasses of wine and in a very good mood, I had a 1-hour talk scheduled. And when I woke up this morning, I had a 45-minute talk scheduled. So we might not get through all of number 3, in which case, well, you're lost. We'll see. If I talk quickly enough, we'll try to do all 3. But so detecting somatic mutations. Oh my gosh. Detecting somatic mutations. So we've heard a lot about this. We had a whole really nice talk on structural variants, these types of changes, which are also very common in tumors. Single nucleotide variants, I'll say a little bit about. All types of aberrations, pretty much everything you've ever heard of happens in cancer genomes, and probably aberrations that you may not have heard of also are happening in cancer genomes. Cancer genomes tend to be highly rearranged. So in terms of the detection problem, it's very similar to things that you've heard before. We take reads, we align them to a reference genome, and then we examine differences between the aligned reads and the reference genome, and then thereby through a variety of algorithms detect different types of changes. So for example, if I pile up a bunch of reads and I see single letter differences from the reference genome, this might indicate that there's a single nucleotide variant there. If I pile up reads and I see split reads or breakpoints between them, this might indicate various types of structural rearrangements. If I see a lack of reads in a certain part of the genome, it might indicate a deletion, either homozygous or hemizygous. If I see too many reads, it might indicate a gain. Again, all of these things you heard about very nicely in the structural variation tutorial that Faridun gave this morning. Now, the one difference in cancer genome analysis is that we are interested in somatic variants. And so in order to distinguish whether we're looking at a somatic or a germline variant, most of the time the experimental methodologies will sequence to match normal sample as well. And this is because we don't have full analysis, full characterization of all the germline mutations in the human genome. I mean, even with the 1,000 Genomes Project, we have only the variants down to maybe 1% or 0.5% frequency, but every individual is different, and we want to make sure to separate out very carefully the germline versus the somatic mutations. So the methods I'm not going to go into in detail because you heard the structural variant methods described very nicely this morning. The single nucleotide variants I'll say just a little bit about because of some of the unique twists, but there's many methods. In practice, people will use methods that are used in any sort of germline study, but there are specialized methods that have been developed for the somatic mutation problem specifically. And let me tell you maybe the 2 reasons why that's the case. The first is that in comparison to looking at normal genomes, germline genomes, cancer cells tend to have a much larger class of mutational events. So they tend to be fairly highly mutated, rearranged, aneuploid, unstable. In addition to sort of this taxonomy of events, there are various classes of complex aberrations that you don't really see in germline genomes. Really clusters of somatic mutations, so-called contagious clusters of rearrangements, so-called chromothripsis and chromoplexy, specific mutational mechanisms like this breakage fusion bridge mechanism, which really results from loss of telomeres and unstable genomes. And so if you really want to detect these types of things, you might need specialized algorithms to do that. And people have developed those, so I'm not really going to go into detail on exactly how those work. And then the second major difference from just sort of plain vanilla variant calling in normal genomes is this issue that we're sequencing mixtures. When we are taking a bulk tumor sample, that sample has admixture by normal cells as well as tumor cells and cells with different somatic mutations. So it's important then to really calibrate the variant calling methods in light of this intra-tumor heterogeneity. So just as one example, if we wanted to call single nucleotide variants, then the classic way to do this, again, is we align the reads to the reference genome. We look for reads that don't have the reference allele. And then, we might count the number of reads and look at the number of reads that have the reference allele and have an alternative allele. And there are, methods that sort of do this. But if we're looking at cancer genomes, a lot of the somatic single nucleotide variants are actually expected to occur at very low frequencies in the population. So we need very sensitive and very specific methods to call SNVs accurately in tumor samples. And so just, there are many such specialized methods. Just one example is on these slides, a method by Nico Bierenwinkel's group where again, this idea of having a matched normal is there. So now you count up the reads that are both in the tumor sample and the matched normal from the same individual. But then because you really want high sensitivity and specificity, you develop a model. And in this case, it's a graphical model based on beta binomial model to really accurately model the read counts and detect mutations at very low cellular frequencies. So you sort of have this very accurate modeling through this graphical model of both the tumor and the normal, and then you use this likelihood ratio test from the output of the probabilities from these models in order to predict whether you have a somatic variant at that locus. But we could actually think of going a little bit further than just detecting the mutations. We could actually try to say something about what the composition of the sample is. So it is very important to know, for a variety of reasons, like what fraction of your tumor sample might be just normal cells and not tumor cells. It's called normal admixture. Which fraction is purity. You'd want to know this number. It would help you calibrate a lot of analyses. And it might also be interesting and important to know, and I'll show you an example in a second, that if there are multiple so-called clones or subclones within your tumor, multiple different distinct populations with different complements of somatic mutations. So there's specialized methods that have been developed to try to infer this type of structure. So could you sort of take just the raw reads from a mixed sample and deconvolve them and separate them out? And this is important because this clonal structure, the amount of intratumor heterogeneity, has been shown to be correlated with things like outcomes. So this is actually old data. There's more recent papers that have shown this, but here you can see that the number of sort of clones, distinct populations that you can measure in a tumor distinguishes different groups of patients in terms of their in this case, incidence of esophageal carcinoma. So you really want to try to get this clonal structure. This is something very different from what you would see if you were doing a germline disease study where there isn't this level of intratumor heterogeneity. So how might we do this? Well, if we can look more carefully at these read counts, both for single nucleotide variants and for copy number aberrations. For all types of variants, you could attempt to do this. For single nucleotide variants, you could go back and you could look at your aligned reads and see that some fraction of them contain the variant allele. And that fraction is actually correlated. In fact, it's, all things being equal without copy number aberrations, directly proportional to the fraction of cells that have that variant allele. Okay? Now, for a single SNV, this is perhaps a very noisy measurement, because if I only had 5 reads covering this position and 2 out of the 5 of them were containing the variant allele, I would, I'd sort of say, well, that's 40% of the cells have the reference allele. That's not going to be a very accurate estimate. But if I started having hundreds and thousands of reads, and then this would become a much more accurate estimate of the frequency. You could do a similar trick with copy number aberrations. Here I'm showing with read depth. So if we had a deletion here, this red segment deleted, then if this deletion was present in all the cells, we would expect a large drop in read depth. But that drop in read depth will attenuate as I mix in, say, more normal cells that don't have this deletion. And the other way of thinking about it is this decrease in read depth is proportional to the fraction of tumor cells that have the deletion. And if you had, say, more than one population of tumor cells, some have deletions, say, in this red region and other regions along the genome, some have deletions in this gray region and other regions along the genome, then these characteristic drops in read depth should be consistent across the genome. So what one could attempt to do then is take these types of signals and separate out, sort of deconvolve the sample into a mixture of clones. And there are different approaches that have been developed for different types of events because of their different characteristics. So again, for single nucleotide variants, each variant that I measure is a very inaccurate and noisy measurement of the frequency because there aren't that many reads that will support a single nucleotide variant, in contrast with a large copy number aberration. You can even think of a loss of a whole chromosome. A loss of a whole chromosome in a tumor cell will perturb millions of reads, so it'll be actually a very strong signal. So sort of two different extremes is, the copy number aberrations perturb many reads, but there are relatively few per tumor compared to single nucleotide mutations where there might be thousands or millions per tumor, but each such mutation is gives you a fairly inaccurate estimate of this clonal frequency. And so you could take different approaches for single nucleotide variants, sort of cluster them by frequency. For copy number aberrations, you could really make use of the fact that copy number aberrations give you these integer constraints. I'll show you two quick examples of that. So for single nucleotide variants, we could collect them all. So I take my tumor sample, I sequence it, I get an estimate of this variant allele frequency for each somatic SNV, the fraction of reads that support that mutation, and then here is just a histogram of those variant allele frequencies. And if I look at this histogram, I might see a sort of grouping of mutations. Here's hundreds of mutations in this cartoon that are all kind of centered around the 60% variant allele frequency. So if I assume that mutations with similar variant alleles frequency are in the same cells, then I can just cluster according to variant allele frequency and produce subclones. And if I do that in a more sophisticated way using uncertainty in the variant allele frequencies from the read counts, then I can get even better estimates of this clonality. And there have been dozens of approaches that have been developed for this that in many ways just adapt existing clustering algorithms. So one example is this method Cyclone. Here's a picture, a figure from the paper, which uses a Bayesian non-parametric model, so-called Dirichlet process clustering model. And what they've done here is they've taken this variant allele frequency plot and they're showing you the posterior probability of the two different clusters here. This is the sort of gray line. Here's the sort of raw data, and you can see very clearly this, I think, a breast cancer sample, that there's two distinct clusters of mutations. Here's a sort of messier sample, is an endometrial sample, where you can see that this it's not as clearly defined into terms of these clusters, but here's at least the model's output of 4 different clusters, and there's some interesting things perhaps that one can infer about mutations being present in most of the tumor cells being mutations that are early in the progression of the cancer, mutations that are present only in a minority or small fraction of the tumor cells being perhaps later in the progression of the tumor. A different sort of approach using copy number aberrations, and this is actually work from my group, former student, a current student, Greta, who's actually here in the front row. And here you use the notion that copy number aberrations give you they come in terms of integer values. So you can, in this cartoon, think of this mixture. Here's a mixture of, say, normal cells that have 1 copy of each of the 3 segments here. Really, this should be 2 because it's diploid, but for simplicity, we'll just have 1. And then you have tumor cells that have 4 copies of the red interval. So you can describe this mixture by just 2 parameters. Each column here indicates the number of copies of each segment of the genome. Think of these segments as being large, megabase, multi-megabase size segments. And so you have your 2 columns describing the two different genomes, and then you mix them in some unknown proportions. And so now I can describe this observed read depth data in terms of just the amount of column 1 and column 2 that are present. And so it's just a matrix vector multiplication, and then it's sort of appropriately normalized to account for the probabilities of events mapping to different places. And then what we want to do is just reverse the process. This is the sort of deconvolution process. So to some extent, this is you can think of as some sort of fancy matrix factorization type approach. If I know there's a group doing matrix factorization in the journal club, so you could take C times mu is what you observe, sort of appropriately normalized, and you want to infer back C and mu separately. Okay, and we have approaches to do this. Here it's based on an optimization approach. It's really maximum likelihood, but you can compute the maximum likelihood using a specific optimization scheme. To give you a notion of how that works, here's data from a breast cancer sample. This was sequenced to fairly high coverage. And what you're seeing here is the individual chromosomes. I'm not showing all the chromosomes, but only those that have some change in copy number. The gray here is the raw read depth sort of pooled over small segments. So you can see very clearly that there's a drop in read depth in the first part of chromosome 1, sort of a loss of first part of chromosome 1 and a gain of the Q arm of chromosome 1, chromosome 3 mostly unaffected, et cetera. And what then the algorithm is inferring is it's taking the signal and it's separating it out into two distinct subpopulations here, red and blue. And what you can really see is that this drop in read depth here in chromosome 1 is sort of ever so slightly more than the drop in read depth here. And what the algorithm then infers is that that's sort of a signal that there's a second subpopulation, and this deletion is only in that population. So what you can see is the tumor, 100%, the sample that we sequence from the tumor, the algorithm refers as 28% normal cells that don't have somatic copy number aberrations, 72% tumor cells, but those tumor cells are divided into these red and blue subpopulations in different frequencies with different events. So there's many other approaches that attempt to do the same type of analysis from copy number aberrations, as well as mentioned, the approaches that attempt to do this from single nucleotide mutations. So going further, beyond just detecting somatic mutations and inferring subclones within the tumor, we could actually try to say something about the process that these mutations, the mutational process that led to the tumor. So this again goes back to this classic paper of Peter Knoll, here shown in this cartoon, where there was some normal cell that got sort of the original driver mutation. This was the founder cell of our tumor, causing that cell to sort of grow faster than its neighbors, outcompete its neighbors, and create a population of cells that form the tumor. Later mutations within this cellular population gave the distinct subpopulations with distinctive advantages. We can describe this evolutionary process, like any evolutionary process, by a tree. Describing the individual cells within the tumor. And so the problem we would have is just the classic problem of phylogenetic inference. If I sequence the tumor at the present time, can I go back and infer the tree that describes this process? So this is phylogenetics 101, classic sort of stuff. And so this has been applied recently in many studies where they've taken tumors and then chopped them up into multiple distinct samples. Here's an example of kidney, an example from prostate tumor. Here's a liver sample. They really went way overboard, chopping it up into 286 distinct samples, sequencing those samples, measuring the mutations within them, and then just applying phylogenetic trees to build, applying phylogenetic techniques to build trees that relate these samples. But actually, if you look at the trees that are published and you think about the analysis that was done, it was an analysis where each leaf is a sample, and we're describing relationships between samples. But as I told you, the samples actually are not necessarily homogeneous because the samples are not single cells. They're still collections of cells. So what that means is that in the phylogenetic terms, we haven't observed the leaves of the tree. We've observed something else. So to sort of emphasize this point, what we really want to construct the tree on is the individual cells in the tumor. Those are the ones that are dividing and acquiring mutations. If we had single cell sequencing at high fidelity, which we're getting there but don't yet have, and certainly we don't have for thousands of tumors, we would then be able to apply classic phylogenetic techniques. So here, an example of just trying to build a tree from single nucleotide variants. Single nucleotide variants, you might have two states, a mutated state and an unmutated state, the normal reference allele. So I could now describe my individual cells by this character matrix or binary matrix, and then apply classic phylogenetic techniques to infer it. But we don't have that. Instead, what we measured were these varying allele frequencies when we had bulk sequencing. So we measured these mixtures. And so you could ask the question, and this is an interesting analytical question, could you reconstruct a tree from not the leaves, but from mixtures of the leaves? It's a very specialized phylogenetic technique, and one that's become somewhat popular to work on. And I'll just quickly show you some of the work that's been done. Okay, so notice here what I did was I replaced this 0-1 valued matrix where I measured individual characters by frequencies. And those frequencies were obtained by some underlying tree, which I don't know, where I've sort of mixed the leaves in different proportions, which I don't know. Okay, so if we try just the simplest possible phylogenetic model, this is something that you might have heard of as the perfect phylogeny model, where characters have two states, 0 or 1, and they satisfy what's called the infinite sites assumption. It's also a popular assumption in population genetics, meaning that a mutation occurs at most once at a particular position. And if we think of 0 as the unmutated state, then what it says is that a mutation the state changes from 0 unmutated to mutated only once on the tree. This is a pretty strong assumption, but perhaps not a too restrictive of an assumption for cancer analysis, because we don't really expect that somatic mutations happen at the exact same nucleotide in the genome more than once in the progression of the tumor. So with this infinite sites assumption, you can actually, this is the simplest phylogenetic model in the sense that you can take the 0, 1 valued matrix and compute the tree according to this infinite sites assumption in essentially linear time. It's the easiest phylogenetic problem there is. If you mix this problem, meaning instead of giving you the 0, 1s, I give you these frequencies of 0 or 1 in each of my samples, then the problem actually becomes essentially one of the hardest computational problems around. It becomes what's called an NP-complete problem, and lots of people have sort of worked on this in the past few years and published different methods that are attempting to build this tree from the different mixtures. Let me just show you one sort of piece of math related to this. So the idea here is that there's this underlying tree here, and under this two-state perfect phylogeny model, you can represent this tree by a matrix where we indicate each mutation is present in each leaf or not with a 0 or a 1. And so there's a one-to-one correspondence between trees and these 0, 1 matrices. And then the observed frequencies are obtained just by mixing these clones in certain proportions. So that gives me this mixing matrix U, and so what we're measuring these frequencies are just this matrix U times this matrix B that describes the tree. F is U times B, and then what we want to do is invert the process and figure out, could we pull apart, could we factorize the measured frequencies as U times B? So it's not a standard matrix factorization problem. It's not just something you can apply PCA to and be done, because there's a very special structure to U and B. And you can figure out what the special structure is, and you can prove theorems about it, and we've done this, and I won't really go into sort of detail about it, but you can actually figure out and prove exactly when you can reconstruct this tree. This actually leads, though, to sort of other interesting computational analytical challenges, because this was just for the simplest phylogenetic model. 0, 1 characters, 0, 1 value characters, where they had this very strong assumption on infinite sites. So now you could ask the question, what if I took any other phylogenetic model? What if I took a maximum parsimony model and then I mixed it? Could I infer the tree back? It actually turns out to be very hard to do this. So even when you go from 2 states to more than 2 states, which turns out to be very important for cancer genome analysis, because we don't just have single nucleotide mutations, we have copy number aberrations that are going to change the number of copies of each part of the genome. And if you mix those and try to pull them apart, then it gets really complicated. We've done this recently, and we have a paper that's coming out soon, as well as actually others in this room have been working on this problem. So there's sort of many interesting things to do here. You can apply it to data, you can get interesting stuff. Oh, good. I do have time for part 3. Great. So now part 3, to some extent, the part that's very important for interpreting cancer genomes, for knowing what's going on, for predicting what are the driver mutations. So what driver meaning causal mutations, so how can we predict which of the mutations that we measure after we've perhaps done step 1 very carefully? Maybe we've even done step 2 very carefully, so we know all the clones that are in our sample, but that's just all measuring all the somatic mutations. Now, which of these are the ones that are going to be important for driving cancer progression? So there are multiple ways people have tried to attack this problem. And to some extent, they parallel approaches that have been used in other studies of other complex diseases with, again, a couple of unique twists. So one is just to use all the information we have about the genome in terms of the annotations that we have, if it's coding regions, in terms of what we know about those protein sequences. And we can attempt to use that information and then prioritize mutations in a certain way. So if we know that a somatic mutation is occurring at a position in a coding region that creates a protein sequence change, and we know that this protein sequence change is an important residue of that protein, or it's in a region that's highly conserved across species, then that indicates that this is perhaps a causal mutation. These types of analyses are done in prioritizing any type of mutation in the genome. You could do this for non-coding regions as well if you have information about where transcription factor binding sites are, where enhancers are. Any prior knowledge you have about the genome you can use to sort of turn into a predictor. And there's a whole host of methods that have been developed to do these types of things. Of course, they rely on a lot of prior information. So you could instead try to use a more statistical approach where you can take the approach that you have mutations from lots of genomes. So within, say, TCGA, we've measured thousands or tens of thousands of cancer genomes. And we could say, well, perhaps we can prioritize by looking for mutations that happen more than we expect by chance. So this is sort of the equivalent of the case control study that you see in GWAS. Again, though, because it's somatic mutations, the controls are the normal genome, right? So we just get to really have the cases, if you will. And what we really then get to do is look at just frequent mutations or frequently mutated genes. So we can see that if a mutation happens in a position more than expected by chance, or if a gene is mutated more than expected by chance, somatically, then we might predict that it's a cancer-causing gene. So just to show you some real data and give you a sense of how this works, here's a data from the Cancer Genome Atlas, over 3,000 patients from 12 cancer types. If you just look at which genes are frequently mutated, here are the handful of most frequently mutated genes. First one, P53, many of you will know. It's the most famous cancer gene around, so the experiment seems to have worked. Second one, TTN, a gene many of you may not have heard of. PIK3CA, MUT16, et cetera. So this first gene looks pretty good. A few of these other genes look pretty good. The others are somewhat suspect, although they are actually interesting genes. Does anyone know TTN? It's an interesting gene, yeah. Do you know what's interesting about Titin, as it's called? It's very big, yeah. By some measures, it's the longest gene in the genome. So, I mean, of course, this is silly, and there's a lot of statisticians in the room, so this is a ridiculous analysis. But you'll see analyses like this published sometimes, not in this particularly silly way, but a lot of these genes are actually outliers in the sense that they're the longest genes of the genome, right? So if we're going to discover frequently mutated genes, we better do it right. And of course, the way to do it right is that what we really have, maybe the simplest model one can use, is that it's a coin flip experiment, right? Each individual is in our cohort, either has the gene mutated or not. If this was random, then there's some probability that they would have a mutation in that gene. Of course, this probability is going to depend on the length of the gene. So if we had a background mutation rate that told us the probability of just having a somatic mutation by chance at each position in the genome, then the probability the gene would be mutated would be directly correlated with its length. And then we could now predict by just using a binomial model and saying now genes that are mutated more than expected by chance, the more genes that have more heads than they're supposed to are those that are cancer genes. And so if we just do this simple model actually on the same data set, we in fact do remarkably well, which is pretty good. So here's P53, PIK3CA, also cancer gene, P10. These are very famous cancer genes, VHL, frequently mutated in kidney cancer. But then we start getting some other stuff that's a little bit suspect. CDKN2A, a real cancer gene. But some of these others are somewhat suspect. So it doesn't quite work out so easily. The reason it doesn't work out quite so easily is that it's actually very hard to get this background mutation rate parameter estimated properly. And it's not uniform across the genome. So our background mutation rate is not equal across the genome for every nucleotide. It actually varies according to things like the replication timing of a gene, the expression level of the gene. It varies across cancer types. Some cancer types have higher rates of background mutation than others. Certainly things like melanomas, cancers that are UV induced have really high background mutation rates due to this environmental exposure. So to get this parameter estimated accurately is actually quite a challenge. And if your parameter is off a little bit, it really starts to skew this model. Interestingly, even in non-coding regions, there's actually variation in this rate. And some of these are actually due to interesting causes. So here's a paper from a couple of months ago where they observed elevated rates of mutation in transcription factor binding sites, which at first seems like a very amazing thing because now you're finding transcription factor binding sites that have somatic mutations, so you're discovering all these causal mutations. But it turned out that the background mutation rate, actually, that one would expect of these transcription factor binding sites is actually depressed because these regions are protected from the DNA repair machinery. So because the transcription factor is bound there, the DNA repair machinery can't access it. This is at least the hypothesis, the explanation for why there are these lower rates of mutations. So getting this parameter accurately is really quite a challenge. And even if we get this parameter estimated accurately and we can look at genes and predict whether they're cancer genes one at a time, it turns out that there's really high levels of heterogeneity across individuals, so much so that if we try to look for significantly mutated genes, recurrently mutated genes, even with the perfect statistical model, what we tend to find is that there's only a handful of such genes that are recurrently mutated, and then there's what's called the long tail of rarely mutated genes. These genes include many well-known cancer genes in particular cohorts, but what's happening, this red line here, is somehow this indicating our level of statistical significance with the number of samples that we have access to. And so this long tail means that we just need really large numbers of samples to try to, one gene at a time, predict whether or not these are cancer genes. So why are we observing this long tail? What's really going on here? And this is having to do with the fact that genes, as you know, don't work alone. They work together in pathways. They interact with other genes and proteins, and regulatory networks, signaling networks, et cetera. So we, to some extent, shouldn't be necessarily analyzing this picture one gene at a time or one mutation at a time. We should be analyzing at the level of the growth pathway or the immune response pathway or things like this. So this is the other level of sort of doing interpretation by looking at these pathways. So you'll see pictures like this in many of the cancer genome studies where what they've done is they've taken well-known pathways here and just layered the mutation data on top and then shown you that although there may not be a gene that's mutated in more than 30 or 40% of the samples in this study, the Wnt signaling pathway is altered in 95% of these samples, so this type of analysis. But computationally, you can go further and you can sort of ask the question, could we discover the pathways? If we have all of this data, and if the hypothesis that mutations, somatic mutations, target these pathways, then shouldn't at some point the pathways start to reveal themselves? So how might you do this? You could, of course, discover these pathways one gene at a time, but then you'll be sort of limited by this long tail phenomenon. You could take a pathway database or a gene set database and just try to look for whether or not sets of genes are enriched using similar statistics that you would use for single genes. But then you won't discover anything new this way, and you won't necessarily be able to look at crosstalk between pathways and interactions between pathways, et cetera. So what has been done is to sort of look at methods that might allow us to kind of dig deeper into the long tail, reduce the amount of prior knowledge that we're using, not just restrict to the hundreds or thousands of sets or pathways that we know, but ultimately try to discover the actual pathways that are being perturbed by somatic mutations. So unfortunately, as we look at all possible sets of genes, that's way too many to test and maintain any statistical rigor because of the multiple hypothesis testing problem. So as the number of combinations grows exponentially. So you could look for methods that are maybe somewhere in between. Is there anything you can do between testing known gene sets and testing them all? And so what's been developed are methods, and these are still very active areas, that either look at interaction networks that are somehow in between known pathways and no prior information, and try to pull out mutated subnetworks that are mutated more than expected by chance, or look at correlations or anti-correlations between mutations across a cohort. There's an approach called mutual exclusivity. So I think in the interest of time, I'll skip the second. I'll just sort of mention the first one. So here the notion is take some representation of the interaction network within the cell. So this could be well-curated literature interactions. This could be high-throughput interactions from yeast-2 hybrid screens. Could be some combination of both. Now take your mutation data, and what you would sort of hope to see, if the hypothesis is true, is that these mutations, the somatic mutations, the functional mutations, are clustering on discrete subnetworks here that are mutated more than expected. So it's these interactions that are being driven. So how can you sort of take these two types of data and sort of pull them together to pull out these mutated subnetworks that must simultaneously consider both the scores on the genes, so like how frequently they're mutated, as well as the connections between the genes. And because interaction networks, biological interaction networks, if you've ever looked at them, tend to be, have a very heterogeneous topology. They tend to have this power law distribution. They tend to have nodes of high degree. This will create problems for this types of analysis, and you need to overcome them. And a number of groups have developed methods for doing this. Here are just two of ours. I'll skip the last approach since we're sort of running out of time here, and just sort of conclude by saying I've only scratched the surface. There's lots of work to be done here. It's a very fast-moving area. I think in terms of analyzing the somatic mutations that cause cancer, it's important not to do it one gene at a time or one mutation at a time, because it's really somehow about all about combinations. But in addition to just mutations, you have to also consider combinations of data types. And because of the extensive heterogeneity of across individuals, you really have to have well-stratified samples. So saying that you sequenced 100 lung adenocarcinomas is not necessarily the right level at which to stratify your patients, and that's really going to start to confound a lot of these analyses. So it's really about looking at the right combinations of patients and those that have the right clinical features, and this is work that's being done now. And then this notion of intratumor heterogeneity. Of course, one individual we can't necessarily treat as just a single clone, because it's really a population of cells. So this notion of intratumor heterogeneity is important, and how these kind of two notions of intra and intertumor heterogeneity interact and how we can analyze the data this way is really work that needs to be done. And then finally, I should say that, of course, as the sequencing technologies change, all the steps in terms of analyzing the primary data, detecting the variants, looking at RNA-seq will change. And as we start to move towards single cell analyses, this will also open up new analytical challenges. So certainly, from the perspective of those of you that are students or early postdocs, I would say that there are lots of areas that are in need of methodological innovation here. So I'll close with just a few references that are also sort of on the schedule. These are a couple of review articles where you can learn more about some of the things that I've described. And I will end there. Thank you.